So thank you, Max and Ruben. I'm following up what Lutz said. This is a traditional meeting, virtual this year, but thank you for the invitation. I see familiar faces going back to my postdoc time, and I was very pleased actually to see Andrea Cavalli listed. That he goes back to my PhD time. So a beautiful group of people gathered today. So this is the title of the talk. So I want to start with kind of a funny slide, and hopefully, I hope that I'm able to. Yes, here we go. So this is a beautiful memory picture, and I'm sure that after these talks, you'll be able to recognize some of the guys on the picture. That was 2018. It was after the same meeting that we have today, and the tradition was that after the meeting, we were going to the rock bottom, here on the bottom, and have a nice dinner. And I heard, sadly, that the place is permanently closed, and so sadly, try to thinking about that we have to find a new place, but definitely lovely memories in talking about science, talking about back in time, someone's getting married, someone getting kids, someone had kids. So this place will be with us forever. So today, so if we go back to 2018, the last time that I had this meeting, I had published a beautiful paper with the National Cancer Institute that was with microbial based interaction with host cells in the gut. But we had just published, and I remember I was able to just show the title, but I was not able to talk about this. And when Andy kindly talked to me about this meeting and the invitation last month, we had just published this paper, and the paper was out there. So I said, what a great opportunity to actually use this time I have to talk about this paper. So Oregon Fluorous enables quantitative intracellular pair agent imaging to assess drug target availability in live cells and tissue. So let me start with showing two pictures here. Summer Gibbs, it's basically an expert on imaging and fluorescence imaging as a bioengineering background. Lee Wang was her postdoc, a very talented postdoc, and I'm not joking. Is doing synthetic chemistry, molecular biology, structural biology, cell biology, and animal experiment. So a very unique individual. And that was really an interdisciplinary work. And back in time was before the pandemic. 2019, Summer approached me at the Knight Cancer Institute. It was like, listen, this is what we're doing. I'm interested in imaging, small molecules. We want to design and develop those pair agent imaging. Can we use structure-based design techniques? And that was the first question. And we went from there. And it was a beautiful collaboration that went on through the pandemic for those hard months and years, as you know. But we were able to get to the beautiful paper that I'm showing today. So let's start with, and I have a little bit of the top part with the thing, but I think you can read the title here. So target engagement, people in drug discovery, they know it's very important for several reasons. So we really didn't invent anything new in terms of concept. This is the 2018 by Promega, very well-known assay. So if you think about this intracellular kinase, you have the nuc, you have the probe. When there's in proximity, there's a Bragg signal. If you co-treat and you compete and you displace your probe, you have obviously decreased the Bragg signal. So it works well in live cells, and you don't have to disrupt the cell membrane. You can use full-length kinase. It can be used in HTS manner. And a tracer concentration, so the level there of the KD, this assay should measure apparent occupancy of a compound for the selected kinase in live cells. So this is based on competition. The question was, what about live tissues? That was the big question, just one step forward. How can we get there? And the DTA, so drug target availability, it is basically defines by how many receptors are available for treatment. This is a paper that Summer published in 2020, where she was kind of defining these type of terms and what iPAI can measure, right? And usually, at least back in time, was measured with untreated samples. So the first thing was the fluorophore, all right? So people were using sort of fluorrhodamine was very popular, okay? And Summer explained me a little bit that the primary problematic was the water solubility. If you start getting into live cells, get into live tissues, so start having the in vivo imaging, water solubility can become a critical aspect. So there's a need to develop more water-soluble fluorophores. And in addition to that, of course, one other thing is they have to account for false fluorescent signal readout, so something that is not that sensitive to the environment. So in the community, what is an ideal fluorophore? So water-soluble, cell membrane permeable, especially if you target things intra the cell, and insensitive to the environment. So in the lab of Summer, they come up with this fluorophore, and I'm going to the chemistry a little bit later in the presentation, but with the overall balance of charge and improved solubility. And it had all the kind of characteristics you can see here, all these marks that can be used to quantify things inside the cell. So we decided to come up with a strategy. First start, okay, with cells, so what we're going to use. Obviously, this is based on the target that you are looking for, right? So we decided to go for A431, is a squamous carcinoma, and ACCA27, large adenocarcinoma. We have plenty of cells there. And for the in vivo, we decided to use those cells to develop our xenograph models. And for imaging data collection and quantification, we did ex vivo, so using the frozen xenograph, they're very stable also, resective after 4 hours of post-system administration. So that was kind of like this, the experimental strategy. So the overall strategy, we decided to go for the EGFR. So this is a popular, as you know, kinase, and the 2 cell lines that we picked express very well. So that was the target there. Tyrosine kinase inhibitors, more than one, as you well know. So there's popular drug out there that you can use, and obviously the 2 things need to be linked. So the second part of the strategy was, if you remember when I was telling you about DTA, right, measuring untreated samples, we decided to add one more thing. We decided to synthesize not only what targets your desired protein, but also use something that you don't want that target you, which is called the untargeted. And we're going to get there in detail now. So a targeted compound and an untargeted compound. And ideally, if you think about conceptually, sorry about that, what you have that the targeted is going to do the job, and then target is going to take care of all the rest, like noise and stuff. So it is the putting the 2 things together that could give you kind of an ideal DTA. So overall strategy 3, that was a tough one. So we decided for GFR. So you needed a drug, you needed a linker, and a fluorophore. So what are 3 things here in terms of what's designed? One is where you synthesize, where you link your linker to in the drug. The second thing, what is the linker that you're going to use? The third is, if you decide a fluorophore, how do you link together the two moieties, right? So there were different questions in terms of design. For this particular case that I'm going to show because of this paper, we already had an idea of the type of linker. We had an idea of the drug. We wanted to use a lotinib, type 1 competitive inhibitor. So there were things already when we started already, but there are examples that I cannot show because they can be disclosed where things were a little bit more complex. So again, targeted compound, untargeted compound, based on erlotinib. Okay, we have a strategy. What was this, the previous success? So the group of Summer was actually already very well known. They published this paper in 2021, was a big paper with Lee as a first author again. So a team very well established that were doing a muscular tissue. Look here, this beautiful image. This is a three-dimensional, okay, fluorescence intensity topographical map. And you can really see is the muscular tissue with its depth. Okay, and usually what is the target back in time for this paper was the brachial part. I don't remember is a particular name, brachial plexus, I think, which is full of nerves, right? Which is 1 millimeter beneath the muscular tissue. And they were using basically on the upper part a visible fluorophore, on the bottom part a near infrared, so NIR fluorophore. See the difference? But look at the beautiful picture on the bottom. So the NIR, which is an oxygen derivative, how beautiful you could see. And what was important there is that this could be used, for example, for very microsurgery. So if you wanted to surgery, it could be a cancer surgery where you want to remove your cancer, but you don't want to damage the nerves. So very, very important for translational medicine. So the group was very well established, beautiful working tissue. So we had all it takes, but we had to start, first of all, again, and I'm not going to show that because that's very for imaging people, but definitely having all this line crossed, right? Yeah, they need something water soluble, so permanent, possess the right photophysical properties, suitable for live cells imaging. Obviously, we won't be here if it didn't work well. So everything was very well done. The bar were crossed in green. And here we go to the chemistry. I always like this, the chemistry biology thing, right? And let's start from the first one. If someone is familiar, I have a lot of people knows a lot today, but you have obviously the part of the moiety of the drug that binds to the hinge, and then you have the part that goes, you know, a little bit in the middle, and then going to that hydrophobic channel of the solvent, right? So the idea was that for the targeted, we were actually going for the part where is going to the solvent part, right? Because you ideally want to keep the hinge moiety part. While for the untargeted part, we decided to block that part, to block the key hinge atoms, right? To make sure that it doesn't bind to your kinase anymore, okay? So the targeted drug and then a targeted drug. And we did a bunch of things. But what was most important thing is on the left part, you had one of the original fluorophore that we're using. And on the right side is actually the optimized, the one that we were really testing and we were seeing all these improvements. And if you see here, there were two major things important. By using this with this sulfur group on the right side, so what you're doing where, okay, I'm gonna keep the charge neutral, but at the same time, I'm gonna replace the carboxylate with a methyl. And that gives you two reasons. You keep certain level of hindrance there, but at the same time, you avoid what's called the cyclic intramolecular cyclization that could decrease your fluorophore properties. So the first thing was, okay, can we predict structure-based design? And obviously, this is a beautiful image. You can see ICM, how well it was able to pick. And if you look at the pictures on the bottom, which are the two new fluorophore, okay, they were very important. What was important was that a sulfonic group that you were seeing before, that was talking to before, making this beautiful interaction with this lysis hub. So everything was aligned very well. And you can see in orange, you can see erlotinib. So everything was very well aligned. And so in terms of docking, things were going in the right direction, especially for 650 and 550. The second thing we do, that was a recombinant AGFR. Obviously, it's important to see the associates, but it's also important to see the dissociates. So the association versus dissociation thing needs to be in a favorable position. And you can see here for the two new ones, the 550, 650, just you should remember this name, things were favorable, right? So the first thing we did, we went to the skin cells, the skin carcinoma, the squamous carcinoma cells. And look at the first thing you do, you look at the target uptake, right? You look at the distributions of your receptors and you compare, right? And you can see really here, for example, if you do targeted and untargeted, things are very good for 60/50 and 50/50. And then the same thing on the bottom here, and you clearly see that you have that targeted intensity versus untargeted intensity. So the ratio is good. You get good uptake, you can keep going up. So we did a step forward and say, okay, now that we have the uptake, we're going to measure DTA. And the DTA, if you remember, two different channels. You got the targeted channels, untargeted channels. You have basically a mathematical equation, and you have the level of DTA. And you can see the big difference from the new fluorophores to the old fluorophores, how nice the nice DTA, the favorable DTA is picked. Clearly a huge improvement with the new fluorophores. So we went in tissues. There was on the left the microsonograph with the lungs tumor cells, and then on the right the squamous carcinoma cells. And again here, beautiful images. On the left here, you can see really the DTA, and also on the right here with the 4381 cells, the DTA also here. And we did something more there for the A431 mice xenografts. Then once we got the DTA, we could treat with erlotinib, and then ideally, if there's competition, you should have decrease of signals, as you can see here. Everything worked very well. But importantly, what you can also do, if you compare, for example, A431 here DTA and ACC 827 mice DTA, you can start looking now how different cell types are behaving. And this is important because usually A431s, they are more rich of EGFR. So also as that confirmed that type of outputs you can get from these type of studies. Interestingly, I use one of the tools with the surface, so the calculation of the surface. It was back in time before we decided to go and using either click chemistry for the untargeted and the classic substitution to synthesize the target and make it the linker. We were actually I try to understand what is the optimal linker, and this is one of the original linker that we're using, and clearly we could see here through docking that that linker was clearly predicted to be not favorable, and also because of the ASAA that give you kind of like low number. So again, here you need to have the fluorophore, which is in a position where there's decent solvent exposure. So that was another interesting thing. We didn't publish that, but it was interesting to see and compare. So this is kind of like one thing from the paper. Clearly, we were successful in improve that 60/50 and 50/50 that have shown substantially improved activity and stability compared to previous fluorophores. Interesting now where we're going for, right? The integration of these two examples into a really bigger platform that open up opportunities to study early therapeutics in cells and tissue with a comprehensive spatial view of drug distribution and binding. And this can be done as a single drug or as a combination. This is kind of like precision medicine. Interestingly, also, when you look at not only drugs but targets, one thing that can come in mind is cancer drug resistance, where you have those different phenotypic effects, different type of these diminished functions. So this is important when you have you want to study cancer drug resistance. And another interesting things that we actually worked already is mechanism, right? And especially if you get those type of outputs, right? It gave you what is your availability, what's your engagement, but also if you couple that, for example, with other type of downstream signaling, so you can start having your upstream target combined with your downstream signaling and see variations at that level. Just some more water. Okay, so OrganFlow platform and structure-based design. So who tells you here already? Interesting, super unique, but obviously, as other many techniques, it relies on decent structures. Okay, it could be structure coming from experimental resolution, but also structure from homology modeling. But have to be different structure. The more far are you from the truth, the harder it gets, just because of all this combination of variables. Having an old ideal together, if you don't have a reliable structure, is hard. So what we're doing, and it's on right now, is testing new EGFR drugs that beyond erlotinib. Obviously, there's drug development, so there's an interest in testing new drugs. But interestingly, also, that's what I cannot disclose today, is other targets beyond EGFR. Okay, so other targets with other drugs, obviously targeting those targets, same or other tumor types. Interested to see the difference, for example, of one drugs, for example, that target two different targets. So the different type of interesting paths here. But we work on very interesting other targets with also very interesting binding modes. And we try also some slightly changes in the fluorophores, but it's all confidential. But what is interesting here, in terms of what we learned, is to design these novel synthesized iPAI molecules for development, is that you have all these questions. How long is the linker? Where is the most favorable linker type? What is the most favorable fluorophore? Where to attach the linker on your drug scaffold? And these are very complicated questions. You want to work all together. And it was really, really impressive how ICM was able to answer a lot of these questions. So we're not talking only about you got the full drug, you dock it, right, and see. No, it started much earlier. It's how ICM is guiding all these different steps to get to the big drug. And I think that was the major accomplishment of ICM for this paper. And for the acknowledgements, I want to thank Ruben and Rita, obviously, always very accommodating, and I know them a long time. So they're also friends. And Andy, let me take the opportunity to today, for the first time, add some wings here, because Andy is probably one of the most gentle people that I've ever met, and he's very patient, very helpful. And I think you're doing a fantastic service to the company, Andy. And that, I thank you, everybody.